It's not actually recording it, is it?
Well, yeah, because I also have to come down right when you start lecture. So we can just usually cut that out.
So it is recording right now?
Yeah.
Okay. So there'll be like 6 minutes of non-stuff at the beginning of the lecture.
Yeah.
So I'll just include a note.
Just saying, many of you guys' mouse is broken. Excluded the first 6 minutes of.
Our lecture starts at 6 minutes.
Yeah.
Actually, what I can do is, this is currently, is this muted right now or not? Oh, it's not on.
There you go.
Okay. Lecture begins at roughly minute 6.
Okay. All right, but everything else should be good.
Okay, thank you. Okay. Let's have a sound check. If we could have a sound check. Can you hear me in the back row there? Can you hear me in the back row there? Okay? Okay. We're already recording, so I don't need to tell the nice AV person that we're recording. Before we start on Chapter 8, do people have any questions about Chapter 7 and enzyme kinetics? Any questions about enzyme kinetics, how enzyme kinetics relates to enzyme mechanisms? Okay, we'll plow ahead then. Let's see, there we go. Okay. So we're going to move on now to another large group of biological molecules. They have a slightly different strategy for making structures than we saw with the amino acids or with the nucleic acids. There's a new handout too. And so we're going to be talking about membranes and the components that make up membranes. And membranes are kind of an interesting right angle to the kinds of things that we've seen so far with proteins and nucleic acids. If we look at the kinds of functions that membranes are going to perform, they're preeminently a barrier. They create an inside versus an outside. And because they create a barrier for an inside versus an outside, they are also responsible for the times, the selected times, the very selected times that things move across that inside-outside partition. So they're responsible for electrical transmission. We're used to thinking of nerves as engaging in electrical transmission. Turns out that most of the cells in your body have similar kinds of behavior. Membranes are going to be the locus for transport, moving stuff from outside to inside either passively or actively. Rather different relationships there, and we'll look at that. Membranes are the locus for many kinds of signaling, of how the outside world can tell a cell time to change what you're doing. And they're also the place in which recognition happens, where populations of cells find another population of cells and go, we're meant to do something together. So in the immune system or during embryogenesis or during wound healing, different cells have to recognize other cell types and do the right thing with each other. And we're going to see that the structure of membranes is entirely intelligible in terms of these functions. That the structure of membranes allows it to be self-assembling. It's inherent in the molecules themselves. There doesn't have to be a directed assembly. Membranes are self-healing, that if you go into a membrane with an electrical pipette or something and poke a hole, the membrane will smoosh back together and seal up that hole spontaneously. And they're also semi-permeable, that they let in parts of the outside world, but not everything. And this is just a very schematic example showing some of those aspects. This is a plasma membrane here showing the sodium flux and potassium flux during electrical transmission. But we're going to see a much better representation of that as we get further into the chapter. So membranes in general are, they're a major component of it is what we call a lipid bilayer. And a lipid bilayer means a structure that's got two layers of lipid side by side. And this is one of the classical representations of it. It turns out that you have hydrophobic compounds and you have hydrophilic compounds. What if a compound can't quite make up its mind that it both loves and hates water? Then it's an amphipathic. Amphi for both, like amphibians live in water and land, or so forth. And pathic, they hate both or they love both. They have feelings about both water and being away from water. And you might want to stop and think about possibly some amphipathic amino acids. But if we look at the structure of a couple of fatty acids, we can see why this would be the case, why it would be amphipathic. It has at one end here a carboxylic acid. This is clearly a very polar group. It's a charged group. It's going to interact beautifully with water. And then it has this hydrocarbon tail, which is very hydrophobic and won't want to interact with water at all. And because the cell has in it a large number of these amphipathic lipids, their structure is such that they will spontaneously assemble in the cell into this bilayer. And it's a bilayer because there's one layer of lipid here and a second layer of lipids there. And they're oriented so that their hydrophobic parts point towards the center and their hydrophilic parts point towards the outside. And all of that can happen quite spontaneously. You can take these lipids in a test tube, shake them up with some solvent, boil off the solvent, get back bilayers. And it's also this spontaneous character that gives it its self-healing characteristic. These are not covalently attached. These are entirely non-covalent linkages. And so if something interrupts that bilayer, they'll just flow back in to reseal because the same chemical properties that made it a bilayer to begin with are still true even after you take out whatever it is that's interfered with it. A self-healing membrane is a really good thing for cells. You know, that you don't need to stop and send an emergency crew to patch it up, but that it can handle that all on its own. We call these two saturated fatty acids because if you look along your backbone, they're carrying as much hydrogen as they can. They're saturated with hydrogen, or they have all single bonds between the carbons. We're going to see that that can vary, that that's going to make a difference to membrane lipids. I wanted to use this figure. This is not, I think, in your textbook. This is from the literature. This diagram of the lipid bilayer is a typical schematic that's given for the logic of how a lipid bilayer works. And it's useful for that reason. It's easy to see what's going on. You can easily see the hydrophilic heads, and you can easily see the hydrophobic interior. So this little sandwich of mayonnaise there in the middle. But it isn't wildly realistic. And this picture is a model at atomic scale of what a lipid bilayer looks more like. It's a little snapshot in time. It should be a movie, so I could show you all these things wiggling and jiggling, but a snapshot in time. And here you can see still the orange business of the hydrophilic heads, and you can see here sort of the gray interior of the hydrophobic tails. But I hope it gives you a sense that it's not quite so rigidly regular as the diagram looks like. It's an ongoing sort of coincidental meeting of strangers. Okay. Now, in our bodies, and in most animals' bodies, there are rarely free fatty acids. Fatty acids by themselves tend to be detergents. They tend to disrupt things. They act like a soap. But more commonly what we see are that fatty acids are part of a slightly larger molecule. They're not part of polymers in the sense that nucleic acids or proteins are polymers that get huge with 30,000, 100,000 molecular weights. These are a little larger, but not a lot larger. And they're not polymers in the sense of being indefinitely big, like a nucleic acid or a protein. And so the way the body deals with fatty acids is by putting it into a few kinds of compounds that keep it from acting like a detergent and make it accessible for the purposes of the body. And here we're going to see one of the ways that that gets handled. Here's a couple of examples of some more fatty acids. This is oleate. It's a mono-unsaturated fatty acid. And there's linoleate with doubly unsaturated fatty acid. Oleate is a major component of olive oil, and oleate literally means oil acid because olive oil was the food oil for many centuries. And linoleate means linen oil. It's from flax, and so it's another plant oil. So these are the free fatty acids. You can see the fatty acids there. And if you were to combine free fatty acids with this 3-carbon backbone, so this molecule is glycerol. It's a 3-carbon backbone with an alcohol hanging off of each of the carbons. So this is the thing that makes many candies stay soft and keeps them from drying up. You can add a fatty acid at any or all of those alcohol groups. And if you do that, you now have a triacyl glycerol. So here what we've done is each one of those alcohols has a fatty acid hanging off of it. This linkage here is a fatty acyl tail. It's not an acid anymore because it's not free. It's a fatty acyl tail. That's an ester linkage there. And so you can imagine that this part of it is pretty polar. That's not so much so, and then the tails are very hydrophobic. Triacylglycerols are really good for storing energy. They're almost pure hydrocarbon. They're not quite pure hydrocarbon, but they're almost pure hydrocarbon. So triacylglycerols are one of the major things that's deposited in fat tissue or adipose tissue. Another way in which we find fatty acids caught up into compounds that make them useful for specific purposes is by taking, again, our glycerol backbone, that 3-carbon backbone, putting on 2 hydrophobic tails from fatty acids, and then doing something else at the third position, what we'll call a polar head group for now. And so this is an example of some of the glycerophospholipids. This is a family of compounds in which there's commonly a glycerol backbone. The glycerol backbone has 2 fatty acyl tails, and then through a phosphate group is linked onto some other stuff. And let's just take a little look at what some of that kinds of other stuff is and the variation that's available to the cell by this strategy. So here's a phosphatidylcholine. This group here is the choline group. And so the choline is linked into the phosphate, and it adds a positive charge over here. Here is a phosphatidylethanolamine. So this is an ethanol with an amine group, so it's an ethanolamine. Again, a positive charge. You can see this is quite a bit smaller than the phosphatidylcholine, so it's the same charge but smaller. Oops, sorry. Here we have phosphatidylglycerol. So onto the phosphate group, we've added another glycerol molecule. This is still a polar head group, but it's not charged. It has 2 alcohol groups, but it only has the charge of the phosphate itself. So net charge on this would be -1. Net charge here would be 0. Net charge there would be 0. And then a fourth strategy is to add onto that phosphate group. This is actually a molecule of serine, right? This is the amino acid serine. And so it's phosphatidylserine. And now we have 2 negative charges and 1 positive charge. So this has a net -1. So we can get a variety of different charges, a variety of different sizes, and so on. The exact shape of the molecule will determine how that molecule can pack. And the polar head group tends to be bulkier or larger than the fatty acyl tail. And so we can see by looking at how the polar head groups are organized relative to the tails what the likely packing is going to be for that group. And so if you look at just a fatty acid, that carboxylic acid is sort of out there compared to the tail. It doesn't really pack very nicely at all. Something that's got 2 fatty acyl tails, like the glycerophospholipids that we just finished talking about, something like that has roughly the same mass in its head group as it has in its fatty acyl tails, and so it packs very nicely into a lipid bilayer. One layer on one side and another layer on the other side with that hydrophobic tails all in the middle. The triacylglycerols that had just the plain glycerol backbone and then the 3 acyl tails, that glycerol backbone is not nearly as big as some of the polar head groups we looked at on the glycerophospholipids. It doesn't really pack very well into a lipid bilayer, but it can just make droplets, plain old droplets of lipid. So this is the packing is the principal reason why the lipids of membranes have 2 fatty acyl tails, not just 1, not so many as 3. Another important group of lipids that are found in membranes are the sphingolipids. Sphingosine is an interesting molecule. This is sphingosine over here, and sphingosine is the common compound that's common to all the sphingolipids. Sphingosine has a chunk from a serine group. This isn't all of serine, but it's a good chunk of the serine. And this is a fatty acyl tail that's come from palmitate. So this gives you a 3-carbon backbone kind of up in here. 1, 2, 3 carbons that we're going to build on. So this is the sphingosine molecule, and here's one of the sphingolipids. The sphingosine molecule is still in black, and here we've added an acyl group of some kind. It might be a monounsaturated fat or whatever. And then on the third position on that alcohol right there, we've added a phosphocholine group, similar to the phosphocholine we saw in the glycerophospholipids. So a similar strategy of having a 3-carbon backbone and 2 fatty acyl tails It differs in what the fundamental molecule is that we're building off of. This is an example with phosphocholine. You also see sphingolipids with phosphoethanolamine. So we've talked so far about membrane lipids like the glycerophospholipids and the sphingolipids that have the two fatty acyl tails and like to make lipid bilayers. But membranes in us, or at least in eukaryotes, not so much in bacteria, have another group of molecules in them that also contributes to the mass of the membrane and modifies the behavior of the membrane, but does not have the capacity to make a lipid bilayer, and that is cholesterol. Cholesterol is your friend. Your body loves cholesterol so much that it makes it every day. If you have gotten into the habit of thinking of cholesterol as evil, I am here to liberate you from that. Cholesterol is a weird and beautiful molecule with many interesting lives that it lives. Well, let's look at cholesterol itself. Here is the fundamental basic cholesterol molecule. And you can see it has these fused rings, and it has a tail hanging off with some branches in it. And this one itty-bitty, teensy-weensy, itsy-bitsy alcohol group. So if you compare all the polar head groups of either the glycerophospholipids or the sphingolipids, they've got all kinds of Christmas trees up there that are polar and charged and all kinds of stuff. Cholesterol just has that one itsy-bitsy, teensy-weensy alcohol. So you can imagine that the rest of this molecule will be embedded in the membrane as far as it can get buried, because all the rest of that stuff is pretty hydrophobic. In addition, this ring region here is rigid. It's hydrophobic, but it's also rigid. The acyl tails in the other two classes we've talked about so far wiggle. You know, I told you that we should really have a movie of them, and they wiggle all the time. This is a flat, rigid structure. In addition to being a bulk component of membranes, cholesterol is also extremely important as a precursor for a number of things. One of the classes we'll talk about a bit are the steroids. Now, in addition to the ones we've talked about so far, the glycerophospholipids and the sphingolipids and cholesterol, all of which have at least the function of being structures. They may have other things as well, but they're all part of the structure of membranes. There are other kinds of lipids as well that do not take part in building the lipid bilayer as a fairly macroscopic structure. So let's take a little bit of a look at some of those. This is a little molecule called isoprene, 5 carbons with a couple of double bonds in there. And isoprene gives rise to a number of different kind of peculiar compounds that have a polymer of the isoprene subunit linking down. And that may have as many as, you can see here, 10 copies of the isoprene backbone linked head to tail. This is the isoprene backbone, isoprenoid backbone for a molecule called ubiquinone. And we're going to see what ubiquinone does soon. It's part of the electron transport chain. Very important molecule. Here's phylloquinone, again with this isoprenoid backbone. You can recognize these backbones with their branches sticking off, right? This is related to vitamin K. It's plays a role in the modification of the proteins in the clotting cascade, called vitamin K for clotting, because it was discovered by German scientists who spell clot with a K. Here's another isoprenoid backbone. This is one retinol. You can guess this is going to be involved in the retina. It also is in the family that gives rise to vitamin A. So it both plays a role in light perception, and it's a vitamin with a regulatory role during development. But again, you see this branched backbone. In this case, it's still unsaturated. The phylloquinone backbone was saturated. This is a non-structural lipid. This one is not derived from isoprene. It's vitamin D. And vitamin D is generated from cholesterol by breaking this bond there. And that bond can be broken by sunlight. So there's a positive role for a certain amount of sunlight. The UV will help you generate vitamin D. To the extent that women in the Victorian period sometimes developed life-threatening breaks in their bones because they would have multiple pregnancies, 6, 8, 10, 12 pregnancies. Each pregnancy withdrew calcium from their bones, but since these women walked around covered up all the time, they didn't make more vitamin D, out of sunlight and would just simply keep withdrawing calcium from their bones until it was to their own harm. So. So I've mentioned that the lipid bilayer is not a fixed structure like a tertiary structure would be for a protein. It's much more of a dynamic structure where things are coming and going and wiggling around and moving around a lot. So the lipid bilayer is really an ongoing average structure rather than a fixed thing like a DNA double helix or a protein tertiary structure. But exactly how fluid it is, how much it wiggles and how much it curves and so forth, can be affected by what lipids are in it. That the particular kinds of lipids will affect how fluid a particular membrane is. And there are a couple of general rules for how membrane lipids affect fluidity. The longer a fatty acid chain is, the more rigid or the higher the temperature it'll take to melt it. Unsaturated fatty acids have lower melting temperatures than saturated fatty acids. So for the same amount of saturation, if it's longer, it'll have a higher melting point. For the same length, if it's more unsaturated, have a lower melting point. And you can see this by just thinking about the lipids in your life, right? You've got butter, you've got olive oil. I hope you've got some safflower oil or corn oil or something like that. The butter can be solid on the tabletop, so its melting temperature is somewhere above room temperature. The olive oil is liquid at room temperature, but if you put it in the fridge because it's virgin olive oil and you like to keep it nice, it'll sort of solidify. And your safflower oil or your canola oil, if you put it in the fridge, it will still stay liquid. It has more unsaturated groups, and so it has a lower melting temperature. It stays liquid at a lower temperature. So that's two components of the membrane that can affect the fluidity of the membrane, the saturation and the length. The third thing that can affect fluidity is cholesterol. Now, if we look at the structure of the other lipids, we can see, I think, why they do what they do. Here is a unsaturated fatty acid. It has a kink in it, and normal, naturally occurring unsaturated fatty acids have cis double bonds. A cis double bond means that the neighboring carbons come out on the same side. That makes them cis. If the neighboring carbons come out on opposite sides, that's trans. So right now I'm a trans fatty acid. And if you look structurally at a trans fatty acid, it's a straight chain like a saturated fatty acid, except it's even less wiggly than a saturated fatty acid because this is a fairly fixed bond. So we can see structurally why those things would affect it. Cholesterol is a little different. Cholesterol kind of slips into or in the spaces between the fatty acyl chains. And the cholesterol has the effect of what you almost might call a fluidity buffer, that it holds the fluidity in a constant amount of fluidity across a wider temperature range than it would be true without the presence of the cholesterol. I also like this picture because it gives you a little sense of the relative dimensions of some of these fatty acids. Okay. So we're getting into fluidity. It is a true and interesting property that lipids diffuse. That is, they can simply move around from the energy of the environment, the little heat energy that gets bumped into the molecules, and they can move around. And they can diffuse in a couple of different directions. They will diffuse from side to side or laterally from one position in this leaflet to further over somewhere else in the same leaflet. That kind of diffusion, the lateral diffusion or side-to-side diffusion, is rapid. It is uncommon for diffusion to happen from one leaflet down into the other leaflet, flipping from one to the other. Now, why would that be? Exactly. The polar head group would have to go through that hydrophobic interior. Exactly. And so that's not going to happen very easily. It might happen once in a while, but it's not something you can depend on. And there are actually enzymes called translocases or flipases that accelerate that movement across that hydrophobic interior. There are some lipids that like to stay together in the plane of the membrane. These are some more exotic kinds of molecules called cerebrosides and gangliosides. And these are lipids that seem to have the ability to stay in little clusters in association with each other. They're sometimes called rafts, lipid rafts, because although they are lipids, they do not simply bump around at random, but they stay in little clusters in the plane of the membrane. And I think I've already mentioned that cholesterol can have this kind of tuning characteristic of maintaining the fluidity of a membrane across a wider temperature range than you would have predicted just from the composition alone. Okay. So, take a look at these these three fatty acids. These are cis-oleate, so you can see it drawn here. It's got them both on the same side. This is a trans-oleate, like that. And then the third compound they're describing is linoleate. These are both 18 carbons with a single double bond. Linoleate is 18 carbons with 2 double bonds. I'd like you to talk with each other and decide which one has the highest melting point, is most rigid, and which one has the lowest melting point. Okay, so chat a little bit and we'll compare answers. What is their relative melting point? Not exact numbers, but relative melting points. Okay. Does anybody feel like you have a relative ranking? Anyone want to take a stab at the one with the highest melting temperature? The one that would be most rigid? Any suggestions? Sorry? The highest melting temperature? Okay. Oh, you want to give an explanation for why you think it would have a high melting temperature? Okay, so you're right in noticing that it has more double bonds than the other two. But the more the double bonds, the lower the melting temperature in general. That is that when we have double bonds, it's hard to pack, and so it's hard to make it solid, or it can stay liquid down to a very low temperature. So linoleate will actually have the lowest melting point. It'd probably stay liquid even in your refrigerator, for example. Okay? So which one would then be the next one in melting point? The intermediate between this and the other one. Okay, you want to say that cis-oleate is, and why would that be? Okay, so you're sort of comparing them both at once, and that's cool. So he's suggesting that the cis-oleate has a lower melting temperature, is more fluid, because the other alternative of the trans is really functionally a straight chain. And you can pack straight things in tighter together than you can crooked things. And that's exactly right. So the cis-oleate's the intermediate in melting point here. The trans-oleate has the highest melting temperature, would probably be in the order of like butter or something like that. And this is why groups like McDonald's and so forth are working to try to eliminate trans fats, is that trans fats appear to be, if anything, even worse for us than plain old saturated fats. That trans fats don't occur in any very great extent normally, and our bodies are kind of going, you want me to do what with this? And they tend to have a worse effect on the body than an equivalent amount of plain old saturated fat. So, you didn't know that there was stand-up biochemistry, did you? All right. So far we've been talking about lipids as if lipids were what you make membranes out of. And you certainly use lipids for membranes, and it's the lipids that make it self-assembling, that make it self-healing. But lipids alone don't make a functional biological membrane. A working biological membrane has to have proteins as well, because the only things that the lipids can do is make this sealing bilayer with a hydrophobic interior that can self-heal and self-assemble. It's striking to think that membranes typically are 50% protein. Most of the diagrams you've ever seen of membranes show a membrane that's maybe 5 or 10% protein. But in fact, membranes have a lot of protein in them, probably close to 50% by mass for many. So they account for the other functions that we talked about: transport, signaling, cell recognition, electrical things, and so forth. All that stuff is the proteins doing things. We've observed 3 kinds of proteins in terms of their relationships to lipid bilayer. You may see diagrams with some other ones, which would be wrong. And these are the ones that are possible based on physical biochemistry. And I'll talk about the real ones first, and then I'll mention the other wrong one that doesn't exist. Some membrane proteins are what we call integral membrane proteins, and they go clear through the lipid bilayer. They have part of the polypeptide exposed on one side, part exposed on the other, and then a continuous stretch of it that goes clear through from one leaflet to the other. A second group of membrane proteins are called peripheral membrane proteins. These are membrane proteins that typically interact with those polar head groups. An integral membrane protein would need detergent to take it out of the membrane because it's embedded in the lipid. But frequently you can get out the peripheral membrane proteins with high salt because it's through these electrostatic interactions that it's hanging on the membrane, and enough salt may be enough to get it off. And then there's a third group of proteins that we discovered relatively recently, which is that there are membrane proteins that have all of their protein mass outside the lipid bilayer, but they have a lipid tail, a covalently attached lipid tail that anchors it into the hydrophobic part of the lipid bilayer. And these are the only 3 kinds of proteins that can exist on physical chemistry browns. You may see pictures with half-immersed, submerged membrane proteins, and those really were probably the confused construct from these lipid tails. There are no half-submerged membrane proteins. So when we think about integral membrane proteins, we have to have some way of getting across that lipid bilayer, both structurally and developmentally. We're going to talk about the developmental part later on in the course when we talk about protein synthesis, but for now let's talk about it structurally. Some of the integral membrane proteins use alpha helices to stretch that distance from one leaflet to the other. And so you have this repeating, coiling backbone of the polypeptide. And in that case, the side chains sticking off of the alpha helix are going to need to be hydrophobic to be in contact in here with the interior of the lipid bilayer. And we can often recognize a membrane-spanning alpha helix because it has a lot of hydrophobic side chains included in it. When you look at the spatial shape or dimensions of an alpha helix, and you look at the thickness of a double membrane, of a lipid bilayer, you can figure out that it takes roughly 20 residues in an alpha helix to span from outside to the next outside of the other. So it's roughly 20 residues from one end side to the other side of a double membrane. This is an example of a membrane spanning alpha helix, and you've got the glutamate's obviously not hydrophobic. Here's a tryptophan, isoleucine, tryptophan, leucine, alanine, leucine, glycine, threonine, alanine, leucine, met, gly, leu, gly, 3, leu, tyrosine, phenylalanine, leucine, valine. Okay, here's a lysine. So here's a charged amino acid. Here's a charged amino acid. There wasn't another charged amino acid in that entire stretch. And a lot of them were quite hydrophobic. So that gives you a little bit of a feeling for it. It's not a fixed sequence. It's more like an average composition. A second way in which integral membrane proteins can get across from one to the other is with what are called beta barrels. A beta barrel is several beta-pleated sheets side by side that are curving around a little bit so they come clear around to make a cylinder. And so you can think of like a barrel, right? If you've ever seen the old-fashioned beer barrels or whatever. It takes a minimum of 8 beta sheets to make a barrel, but that's a minimum. Some of these barrels have as many as 20 beta-pleated sheets stretching around to make very huge cylinders. In beta barrels, there's slightly different structural constraints. Obviously, the residues that are facing out into that hydrophobic interior of the lipid bilayer, those residues have to be hydrophobic to stay in contact with them. But the residues on the opposite face, on the interior of the barrel, those don't have to be hydrophobic. So half of the residues need to be hydrophobic. The other half are not constrained that way. And a common feature that we observe is that at the edge of the barrel, there are often these aromatic side chains. You can see them diagrammatically here, right? These are tyrosines and phenylalanines and tryptophanes. And you can imagine that having aromatic side chains at the top and the bottom help to anchor that barrel in the lipid interior. Because it's only the outside side chains that need to be hydrophobic for a beta barrel, it can be harder to identify beta barrel sequences just by primary sequence information alone, which takes a larger number of amino acids to get, or amino acid residues to get across a membrane, an alpha helix or a beta pleated sheet. Why would that be? So you're adding up all of them together. In other words, you're saying that the whole stretch of the protein. So if you counted each turn of it. I'm just thinking of a single span. So in other words, one beta sheet or one alpha helix to get across. Okay? Would that change your answer? Remember that an alpha helix is coiled. So it takes 3.5 amino acids residues per turn. Whereas the beta sheet you can think of as having pulled the peptide backbone out as far as it can go. So you can get, it takes close to 20 residues in an alpha helix to get across. It will take substantially fewer to get across with a beta sheet. So I mentioned the integral membrane proteins. I mentioned that there are some lipid-linked proteins. And these are some examples of, oh my goodness, that's ugly looking. What happened to that? Whatever. So these are some of the kinds of tails that proteins can have covalently attached. A myristyl group, which is a simple fatty acid. A palmitoyl group, which is related to palm oil, not too surprisingly. A prenyl group. A glycosyl phosphoinositide, which is a little bit funny because this is something that's actually linked with some sugars in between the protein and the lipid itself, so that the actual chemistry of it can be somewhat complicated. Now, you've probably heard of the phrase, the fluid mosaic model for the membrane. And I want to be subversive and suggest to you that the fluid mosaic model might be a good model for lipids, but a solid mosaic model is probably a better model for the proteins. And I think it's a little sobering when you realize that the classical diagrams of the fluid mosaic model, many of them you can trace their intellectual history back to the very first paper in the early 1970s. And you'd think that in 35 years we might have learned something else about structure, which we have. But it doesn't always make it into the textbooks. So that's why I'm here. Lipids do diffuse freely in the plane of the membrane. That is, they diffuse freely side to side with the restrictions of what I mentioned in the lipid rafts. So talking about it being a fluid mosaic model is a reasonably good model for the lipids. The proteins, it turns out, if you actually watch the behavior of the proteins, are somewhat different. In general, proteins are going to move more slowly than diffusion alone would predict. That is, we can figure out the viscosity of the membrane from the behavior of the lipids. We can predict from that viscosity how quickly the proteins move, and the prediction breaks down. There are some proteins that do not move at all that we can measure. And we have various ways of showing that those immobile ones are in fact associated with the cytoskeleton in addition to the membrane itself. There are other membrane proteins that can move around in an area, but not in general. They are constrained to some extent of where they can go. And then there are others that do move, but they might move at 1% or a few percent the diffusion coefficient of what you predict for the viscosity of the lipid bilayer itself. This is showing you sort of a fairly classic fluid mosaic kind of model. And you notice in this fluid mosaic model, there's no cytoskeleton. And all cell membranes have cytoskeleton underneath them, except for the patches of dying cells called blebs, where they have little bubbles that are free of cytoskeleton. One feature that this picture does bring out is you notice this kind of cactus structure that they've got on the proteins on one side. And these little yellow cactus things, sometimes on the lipids, sometimes on the protein, are a reminder that there are sugar residues that are on both the lipids and on the proteins to variable extents in the extracellular side. That all the sugar components are on the outside side of the cell membrane. A slightly more realistic model of sort of the transmembrane proteins and the cytoskeleton, or the integral membrane proteins, is from this picture from a red blood cell. You don't need to worry about the names of the proteins. These are just examples of the kinds of proteins that exist. Now remember I said that 50% of the mass of membranes is proteins, and then figure out what fraction of the mass is being represented as proteins here. You can see this is still really underrepresenting the presence of proteins. But the other things that they have to say here are quite true. And so they're showing you that here is a transporter that is anchored to an actin microfilament here. Here is another transmembrane protein that has a different kind of anchorage into the cytoskeleton, and so on. And so we're beginning to put together some of the sort of a zoo of the different things that can interact between the membrane per se and the cytoskeleton, and the two are actually kind of tied to each other in many points on the inside surface of the membrane. And we can actually observe some of these constraints. This is looking at a transferrin receptor, and they've been able to put a tag on individual molecules of the receptor and track that individual molecule over time. And so what you can see is that for this period of time, it bounced around in this little patch, and then it sort of jumped over the corral fence and bounced around here for a bit, and then it jumped over into this little patch. So it moved around in a patch for quite a while before it made itself over to an adjoining patch. It didn't just simply have a random walk freely all over the membrane. And this is for something that actually does move around. The things that don't move around obviously would be much less exciting. This is another representation. One of the reasons for including this in addition is it's emphasizing the fact that there's cholesterol in there as well as the glycerophospholipids and sphingolipids. The carbohydrates are kind of prettier. They look more like carbohydrates here and less like cactuses. Again, making the emphasis that the glyco residues are all towards the outside. At least it came through this time. It sometimes comes out as vanishing. The amount of sugar on the outside of the cell is pretty formidable. And there are glycoproteins, where you have sugars on asparagine or serine residues. There are glycolipids. I mentioned already some of the glycolipids earlier when we were talking about the composition of these things. But the amount of sugar out there, it typically doesn't register on it. And this is an electron micrograph that I like because it's used a stain that makes sugars show up in the electron microscope. And this little layer right down here at the bottom, right along there, that's the lipid bilayer. And this forest of stuff, this jungle of stuff, is the carbohydrate that's decorating the membrane lipids and the membrane proteins. These sugar residues can make a huge difference. If we look, for instance, at blood types, I don't know if any of you have had to receive a blood transfusion. I hope many of you have donated blood. One of the major questions that they'll determine is your blood type. Are you A or B or O? And it turns out that blood type is due to differences in sugars. So individual A who has a type A blood sugar has modifications on their cells that include this particular sugar, and therefore they raise antibodies against type B. Type B people have this sugar on theirs, and so they raise antibodies against type A. If A people get A blood, not a problem. If B people get B blood, not a problem. Type O people don't have either sugar on there, so you can safely give type O blood to people who are type A or type B, because there's nothing there that their antibodies would react with. There are a few unfortunate individuals who have both kinds of residues on their sugars, and their blood can't be given to anybody, because A people will react to it, B people will react to it. Okay. So far we've talked mostly about structural aspects of lipids and membranes, and now we're going to talk about some more of the functional things and how they accomplish some of the things we talked about. And certainly one of the major aspects is transport. You may remember back to when we were talking about the E. coli genome, about what a big pie wedge was involved with transport. It's not quite as big for humans, but it's a good-sized one, and we'll see why. Things can enter the cell in a couple of ways. They can simply passively come in. That is, there's more outside the cell than inside the cell, and so they passively come in. Or the cell can invest energy to bring it in. That's called active transport. Passive transport can happen, again, in two possible ways. You can have simple diffusion, where a compound that can slip through the membrane just slips through the membrane and comes into the cell. That's just simple diffusion or passive diffusion. There are obviously compounds like sugar, glucose, that are not going to get through that membrane spontaneously. And so you can have transmembrane proteins, integral membrane proteins, that are channels that permit the movement of things down a gradient. If there's more outside the cell than inside the cell, it will flow into the cell in what's called facilitated diffusion. But if there isn't more outside the cell than inside the cell, and you really need it, it may be worth investing some energy to get it to happen. In that case, we're talking about active transport, and there's some very cute tricks that the cell uses in order to generate active transport, but it allows the cell to accumulate a gradient of things inside versus outside. And I think that's kind of a logical place to stop for now. Any questions before I see you all on Wednesday? AV guy, we're done. Yoo-hoo! It's available on Icon, and it's also available on the links page.
Okay, links page.
So there's a toolbar. There's a toolbar at the top of Icon, and links is all the way to the right.
Okay.
And that takes you to the Google videos. Are you on a Mac?